Okay, so now we'll open this up to discussion. So let me just take you back to the beginning, which outlines the topic areas that we proposed. And so comments, questions?
Including things that weren't covered under these.
Yes, thank you, yes. Including things that are not covered in particular. Trey.
Okay, thanks, Jonathan, for putting together a really nice organization. And I want to talk specifically about the trans regulatory networks. First of all, I wholly second or throw support behind that goal as a resource that this institute could uniquely generate. I wanted to make a suggestion about the approach. So the concern would be if you just rely on molecular perturbations followed by profile measurements, many of those interactions are going to be completely indirect. And so one suggestion for additional data that could really be useful there gets back to being suggestion of direct interaction measurements with, say, protein DNA or DNA interactions to complement those perturbational types of studies if the goal is to look at networks. And then, of course, if you're interested in effects outside of the nucleus, then you're talking about following up those with, say, protein-protein interactions. And certainly in our own work, I think in many others also in the cancer mutation analysis field, you find that you really need these direct interactions to knit together how different variants or somatic mutations influence the same subnetworks, if you like, or protein complexes or organelles. And just the perturbational data hasn't been as informative.
Thank you.
All right, so Mathieu Lupien again from the Princess Margaret. So I have 2 points to raise. So the first one, which is like very nice work, but at the same time, I hear a lot of gene-centric focused analysis, and I would like to put forward this notion that the genome can be partitioned in many different ways, not just gene-centric. And so if you partition the genome, for instance, based on cystromes, right, the sum of binding sites for a given transcription factors, and you look for a burden of variants within those, you'll prioritize some cystromes over others in a given disease site as being burdened by variants, and that points out to a function towards variants affecting these TF. And if it's a burden genome-wide, then it doesn't have to be affecting a single gene. It can be that in that process, it changes the equilibrium for that transcription factor such that it suddenly binds more to certain sites, or it binds less to other sites because it's been disrupted globally at a collection of sites based on variants. So I think it's important overall just to consider partitioning our genomes in a different way than just on a gene-centric perspective, because that can still give us a first layer of information that's going to be highly informative. The second point, with regards to the G and E relationship, so the environment, maybe following up on the first comment, a suggestion would be, from my perspective, to actually prioritize environmental exposure that has to do with metabolism, since there's a very close relationship between epigenetic reprogramming and metabolic adaptation. I think that if you want to be able to have a rapid assessment of how environment can influence genome function, to be able to focus on those that are most likely to affect the epigenome, it was a good way to start.
Thank you.
Just to respond to the first one. I was going to say that I think that's a great idea, not just to use variants as a way to prioritize non-coding regions, but to go beyond just human genetics and use things like transcription factor binding sites. I think that can answer questions about like cooperativity. Can a single transcription factor binding site have an effect on a gene or on other things? What's the logic? How does that work? So I think that's a really nice idea about the cystromes.
Great, thanks. Eric.
Yeah, so this was brought up before, but this idea about what is a gene and so forth. And I think that's also going to cause a lot of problems if you try to have a goal of functionally characterizing, doing something to all genes, where that concept may not really exist in a clear format in the actual real biology. And so, the exons plus the introns, they vary from one person to another, and they're splicing and so forth. And you get even fusions of some exons between genes. So, even I use the gene word there. So I think maybe a more useful way of doing about this may require redefining of what that means, and that redefinition could be of a particular sequence of the particular sequence within a certain range of divergence in the genome, what's its function, whether it's a particular protein domain or an entire protein coding sequence or something else. And coming up with a new term maybe to describe that, and then say we'll describe all of the genomic character of the genome or just this part that makes proteins or otherwise.
Okay, thank you. Sorry, I can't see who everybody is from this distance, but...
Me? This is Carrie Getz. I work at the National Eye Institute.
Okay.
And,
I should work on my eyes, sorry.
Oh, that's okay. One of the... Maybe get some glasses.
Yes.
To the last point that you made in your introduction, when you're talking about combining data sets and how you need standards, and one thing that I've been involved in at the NIH for many years is kind of building on what Phoenix started with the Common Data Elements Repository. And I'm happy to say that the NIH has a data science strategic plan that they've put forward. And as part of that, our Common Data Element Task Force that's trans-NIH, so all the ICs participate, did propose a master reference dictionary resource that the NIH would fund on all levels. So I think I would recommend the NHGRI get behind that effort and maximize the collaboration there. And our scope of work says that should be launched in a year for the first iteration. So.
Yes, thank you. So I think another maybe issue we could bring up in that same vein is investigators adopting those standards. So Phoenix has been around for several years, and it just for a variety of reasons, people just don't adopt it until you're faced with, I can't combine my data with yours because we don't have things labeled the same way, or it's difficult to find out if how you measured smoking is the same way I measured smoking. Yep. Yeah,
I would say that a lot of the ICs have found success in writing it into their grants and making it mandatory. So that's
so the carrot and the stick approach.
Exactly.
Yeah.
But once you can combine data and show that efficacy, then I think you're going to be smooth sailing. And if there's a central place people can look, make it easy, that's another component. So that's what we're trying to accomplish there.
And I would add to make it easy also for data repository or data sets that have already been deposited in something like dbGaP. I know Phoenix was going back and trying to map back. And so if there are more concerted efforts to do that, because anything that makes it easier on the investigator end, that'll make the combining of data sets and analyses and subsequent discoveries that much faster.
I would just, this is my own soapbox, so I'm speaking on my own behalf, but mapping back is a poor scientific
no one wants
effort.
to do it. It's not glamorous.
I would say it doesn't even result in statistically significant data, so I totally think you should do the work on the front instead of mapping back later.
Yeah.
But that's my own little caveat. So.
Yeah, and so there are other things I'm sure we had to discuss, but I just want to say that it is a shame that we have so much data already collected. It is so hard to collect exposure data on humans, especially at with cohorts. There are very few big cohorts being funded right now. So mapping back still, I think, has utility because we have so much data. That's just my 2 cents.
Okay. I'm sorry, I can't see your names. Yes, you. I can't see your names.
Stephanie Hicks from Johns Hopkins. So going back to the problem way back on overcoming cultural divides and silos and training to tackle many of these important problems that you have here and the ones that we've been discussing the last 2 days, it can be, I'm a quantitative person, it can be daunting to listen to jargon in a field that you're not necessarily intimately familiar with. So for example, genetics, and it can also be daunting vice versa. It can also be daunting for a quantitative person to go seek formal training in genetics just to be able to understand that and vice versa. So one way you can address that is collaborations. That's more of like a long-term thing. And one recent experience that I had that was very positive in terms of overcoming those cultural divides between genetics and quantitative people or biological people was innovation labs. So it was a project, it was a week-long workshop funded by the BD2K in which they brought together quantitative people and biology people. They were all assistant professors, and they all had sort of projects in mind that they wanted to discuss, and they all came from very different perspectives with very different training, and we quickly were able to overcome some of those cultural divides, and it forced us to sort of work together and put together projects and ideas on how to overcome the cultural divides and then make or propose solutions to the problem. problems. So I would love to see the NHGRI fund more of those. I thought that was very useful.
Just to follow up on that, that's a great suggestion. This was also actually brought up in the ENCODE meeting that we just had recently of trying to get more computational people in wet lab and vice versa, so people can really understand what they're doing and collaborate better.
And I do want to just put in a plug. So we have talked a lot about diversity and participation in research, not a whole lot about diversity in training, and beyond the T32 or other mechanisms for postdocs and pre-docs, but really reaching into undergrads to excite them about our field because we do cool stuff, and I just don't think they're exposed to it, and they'd be really amenable, I think, to the kind of learning about collaboration early on in that team science approach. So I would encourage everybody to think, whether it's at the funding level or even as an investigator participating in something like ABRACAMS. I know a couple of you here have been speakers there, but the systems and computational biology section is the smallest section at ABRACAMS, and I think we could do better.
Great, thank you. Tim.
Yeah, Tim Rennie from Duke. So a lot of these areas, we keep bringing up technology development. And one thing that comes to mind for me a lot is getting technology development funded through NIH can be really challenging. And I think a lot of it has to do with the exploratory nature. When you get into, not quite sure what the results are going to be, it's hard to write grants around that. I think a lot of it has to do with the timing. Oftentimes we've wanted to develop a technology, and by the time the grant is reviewed, we've already, finished the paper, and so the grant review is so much slower than we want to push the technology. And so I'm wondering if maybe over the next 10 years there's ways that NHGRI can think creatively about how to more efficiently fund technology development. Maybe it's smaller grants with faster review cycles. Maybe it's I think it has to go beyond just calls for specific R01s. I think that's one way we could, really skyrocket and lead the field in how to fund technology development at the government grant level.
Okay, thanks. Barak?
Yeah, I wanted to just say a word about phenotyping. I'm really excited about the high throughput phenotyping, molecular phenotyping, things that are scalable. I think that's all great. I think we also should measure success by how much these technologies diffuse out into the wider world. So for example, if large collections of variants and the methodologies to make those variants diffuse out into the wider world, then we outsource our phenotyping to everybody. And we get more. We don't just get RNA levels or the things that we're good at measuring. We take advantage of people who have spent their entire lives developing one particular system. And I think the other thing it does is it circumvents some of these discussions about what's the right model. Should it be cell lines? Should it be tissues? Should it be organisms? We just let the wider world do it for us.
Okay, so Tuli?
Yeah, so I wanted to comment on the cis-regulatory effects of variants. So obviously during the past 10 years, cis-EQTL mapping and other sort of cis-regulatory variant mapping in populations has been basically the primary high throughput workhorse for doing these things. And now we have GTEx and all of these big maps, and I basically wanted to share a couple of results coming out of those studies, and like thinking if that study design is actually useful going forward. And one of the things that is coming out of GTEx, now that we have sample sizes in many hundreds, in many tissues, is that when we do computational deconvolution of cell type composition in those tissues and start to map genetic interactions, so basically kind of cell type interacting EQTLs, we actually gain a lot in resolution. We're able to do better fine mapping. We get to the causal variants better. We're able to do better co-localization of GWAS findings. And we also see that basically a lot of the tissue sharing or tissue specificity of regulatory effects is basically just sharing of cell types between different tissues. And I mean, this is not really surprising that when you take a bulk tissue, your EQTL mapping there is just smashing a bunch of independent signals from different cell types together. But I think that is, it makes interpretation of some of the current data a little bit more complicated, but thinking about cell type specific EQTL studies, I think can still be a very, very worthwhile study design to think about going forward.
Yes,
now that we actually have single cell technologies that, that will soon be scalable to population sample sizes.
Right. Yeah, Eric.
So in addition to all the amazing content, there have been a couple of suggestions about mechanisms. I think it would be interesting to ask, Are there mechanisms that would generate large data sets and make them available as challenge problems to the community? We have a model of build a consortium where we build a lot of data and then everybody gets together and has a very large number of very satisfying conference calls and eventually writes papers about it. But it'd be interesting to explore models of generate some well-defined data sets, get them out there as challenge problems, and who knows, I'll just make crazy suggestions, invite computational people from all over to try to attack them. And some of the rewards could be you get a grant if you have the best thing. Because it might encourage people who produce really good work but might not review really well at study sections or other things. Obviously, we can't switch everything to that, but it might be interesting to ask, are there models that could both draw in the community and reward people in unusual ways? There are probably better ways to do what I'm suggesting, but I think there's a chance to experiment with models here that this might be a really good time to try.
That could also work well for what Tim already mentioned, I think, with the tech development being faster than the grant cycle.
Yeah, that's actually what made me think of it, was Tim's comments about the tech. And I think computation may have the same set of issues.
Yeah. Great, thank you. Anshul.
Yeah, I just wanted to follow up on Eric's comment about challenges. So I think one of the ways to actually do this effectively is to get community buy-in in the challenge design itself. A common mistake that's made, I think, in running these challenges is some external party designs a challenge based on what they think. And when the challenge is proposed, the rest of the community completely rejects the challenge design itself. And to me, having looked at Dream and KG and others, which are really nice frameworks for designing challenges, the one mistake they make, and we have made as well, we ran an ENCODE challenge where we think we should have, we missed a small little wrinkle in how to design the challenge. It was otherwise okay, but I'm just suggesting that we should have calls for challenge design. We should have community agreement on challenge design, then collect the data and that will have community buy-in so that the participation increases. Otherwise, just have very small population of the actual research community playing in those challenges, which is very different from CASP and many other events.
Okay, Mark and then Stephen.
I just wanted to follow up on the 2 points very quickly. I'll just say in relation to challenge design, Anshul did mention CASP, and one of the really nice things about that challenge, in addition to the participants potentially being able to maybe write a grant, there's also just the idea of publishing the results. I mean, I think that challenge has become a nucleus for people working on that particular problem in terms of publishing the results and also people putting in data. And the second point was in relation to Tuli's comments. I concur very much. I want to amplify her comments. I mean, I've been involved in a brain study, and we found that coupled single-cell data was extremely useful to deconvolute a lot of the effects. In fact, the bulk of the EQTL effects were actually cell-type proportion effects.
Mm-hmm. Yeah, thank you. And that also relates to the point that Aviv made in the previous session as well. Yeah, Stephen.
Yeah, I just wanted to follow up on the challenge model. This is something that's being looked at in some other institutes. For instance, NCI, we are developing right now a challenge for mutational signatures where we would put 2 or 3 very large data sets in the cloud. Whoever wants to come bring their model, they run it. And again, this is this issue of bringing the analysis to the data so everyone else can see what the performance looks like, not only the outcome, but be able to compare and learn from that to improve and work on the code or give suggestions or whatever, because often different people coming from slightly different perspectives will highlight some very interesting things in the data set, and there isn't necessarily a single right answer, but those comparisons allow you to start to see where there are differences that could be incredibly revealing for mechanistic or biologic or a computational solution. So this is going forward, and I think as Eric had mentioned, there may be rewards in terms of funding or whatever. That's a tricky thing that NIH is having a hard time figuring out how to do it, but our current director, Ned Sharpless, is very keen to do this in several different cancer-related venues. Okay.
Yeah, and just to follow up on that, I mean, we haven't run the challenge particularly at NHGRI, but we do fund them, and you can actually, if you read, for people, this is partially an advertisement for people on watching the webcast as well. If you read our genomic resources, it actually allows proposition of challenges, and we did that in collaboration with NCI. I do think, to Anshul's point, I think the challenge is doing good challenges, and so thinking about how to do that. And I would love, and I love the idea of giving a grant, but then my head explodes at the idea of that being something that we could legally do at NIH. But I think thinking about meaningful rewards that might not circumvent our entire grants process would be an interesting thing for people to think about, and we would certainly be open to feedback on that.
Thanks, Carolyn. Gil in the back.
Yeah, I love where this thread is going. Just a tiny comment. I think Anshul mentioned it yesterday, right? The hardest thing about a challenge is finding a referee. So one thing that other communities do, they put out benchmarks. They don't call it a challenge. They just say, here's a large data set, see how well you can do on it, and people offline go and try to bite it off as much as they can. So that has disadvantages of its own, but the main advantage is it's there. Anybody can play with it anytime, and you don't need a referee. You just need a good set. We could do it tomorrow with variant, pathogenicity prediction. You just tell people, these 2,000 genes with all of their variants, do not train on them, only test on them, and let's see how well you do. That's it, simple as that. So challenges are wonderful, but you can add benchmarks and then you move much more quickly, and other communities thrive on that completely.
Thank you. So our committee's charge is to come up with a list of precise topics or study areas for tomorrow. And so I want to kind of bring the discussion back to this, whether these are the right 5 topics, whether there are topics you think that we should be adding or deleting, or whether these should be modified. Sharon.
I would just say for the G to E, to go back to something that Dana said, NHGRI doesn't have an epidemiology branch. So we haven't done a lot of epidemiology. The other institutes do. So you don't want to design a project that says we need to learn how to do and to measure things that other groups already know how to measure. I would just echo, when you're thinking about projects, there are a lot of existing data sets with good epidemiology, but have very poor genetic evaluation so far, maybe a SNP chip, and that's it. So I do think thinking about some G to E studies using well-phenotyped environmental exposures that might be from NCI with regard to smoking or NIEHS might be one way to think about projects, and not defining how do you measure phenotypes.
Thank you. Howard.
Howard Chang from Stanford. I want to highlight the Project 3, the trans-regulatory effects. This is something that's clearly within reach with many recent technologies to do so even in single cells. And the really exciting aspect, I think, really comes from the combinatorial aspects when you think about regulators that have, synergistic or buffering effects. Basically, one rescues the other. That's what you want to know, right? If you have this defect, what would it take to, let's say, rescue the phenotype? But then the complexity of the combinations actually will get big very fast. For example, you think about perturbing every link RNA or perturbing 2 link RNAs. I challenge you to decide which 2 link RNAs should go together first, right? This is not a solved problem. Or let's say 2 link RNAs plus 1 TF. Okay, which ones are most interesting? And the space gets, so I think we need to learn a lot more to even thoughtfully build these kinds of studies.
Yes, so your point on the interactions is a really good one, but I would argue that we don't even know very much about marginal effects yet. And there's, so for example, perturbations with 1 gene at a time are already going to dramatically increase our knowledge, although your point on combinations is important. Were you going to follow on for that, Aviv? Okay. And then I'll come to Andy.
Briefly. To follow on that, I agree we don't know the marginals, and I definitely agree with Howard, we don't know how to choose which ones interact. But because interactions are actually pretty sparse, in all likelihood, you can probably just squeeze a lot more information by perturbing a lot of things at once for your marginals and his interactions at the same time. So I think saying let's go for interactions after we figure it out, each of them individually, is a pity.
Okay, thanks. Andy.
I guess it depends on how broadly you define trans-regulatory effects. It could be everything else, but there's nothing here, and there's a lot of discussion yesterday on the effects of repetitive regions of the genome on gene function.
Yes, thank you. Good point.
Jonathan.
If we run, oh.
Hi, Brittany Lassane from Hudson Alpha. I just want to underscore and piggyback on what Tim said earlier about technology development and how that needs to happen quickly. And I want to throw in and amplify the idea that bioinformatics has to be part of that and not an afterthought. We're talking about generating a ton of data here, and in order to move from a catalog to actionable metrics, things like mechanism, we're going to need new bioinformatics approaches to do that. And so, in particular, things that we've talked about like imputation, transfer learning, those sorts of things. So I just want to amplify that before we close.
Yeah. Thank you. Great point.
I have a specific suggestion that I just want to make. The people here on this committee know this suggestion because I made it to you guys yesterday, which is that just very specifically to what Tim said is last year or maybe the year before, there was a somatic cell editing consortium with a whole bunch of funding opportunities. I think this is from the Office of the Director. I'm not sure exactly what part of NIH it came from, but maybe NHGRI could consider something similar since so much of this depends, this is totally in my own personal interest here, but so much of this depends on new genome manipulation technologies, that there could be something similar for these goals, for this kind of variant interrogation, specifically technology development grants that also includes ways of integrating these kind of very large data sets that's completely absent. So that might be something to consider.
Thanks. So I'm astonished that we may have exhausted the comments. So we're a few minutes early, but I think if there are no objections, we can
as long as everybody feels like all their ideas got voiced.
Last call for questions and comments. Okay. Yeah. Thank you. Do you want to? Before you go, I think do we have any wrap up?
So we've given instructions to the topic leaders for putting their ideas in for presentation tomorrow during the prioritization discussion. And if anyone has any questions, we're here. And Adam, do you want to say anything else?
Yeah, I do. And I just want people to, I'm a little worried that in the division of the topic discussions between today and tomorrow, this is general for all of them, that if you're waiting to hear ideas that you think are important and you're anticipating, if you can reasonably anticipate that they're going to come up in the other two topic sessions, that's fine. We can deal with them then. But if you look at the other two topic sessions and you're really worried, now is the time to raise them. And I'll raise one that doesn't need extensive discussion that I haven't heard. At some points, I heard a lot of enthusiasm for better assays for measuring the more difficult parts of the genome to measure. And it didn't come up as strongly as I heard it in other places. And I didn't hear it as much in either of these two sessions today. So I'm asking, especially these two sessions, as you go back and think about what to include, that you consider how that might be incorporated, because I did hear it pretty strongly. That's controversial. People should react. And if there's anything else like that people haven't heard and are worried and are ideas at the level that was presented here, you should at least shout them out so the two groups are aware of them and can think about considering them. And we've got like 4 minutes to do that. So.
Well, Adam, I confess I've gotten confused. So we had reports back from breakout sessions, and we have lists of projects and that have a support for that. Is the process we're going through now the filtering on those? And if what we'd suggested before didn't make it onto this panel, we're supposed to say we were serious.
If you're really not seeing it, you should say we're really not seeing this. And if it's
there's a bunch of them.
Level.
Yeah, there's a bunch of them.
As one of the topic leads for tomorrow, and I can say it's not on our list, we really need to be pointed to those specific summaries from the breakouts, because we're just pointed to this whole Google Drive with lots of notes. I think those breakouts would be particularly helpful for us.
We can do that. They are on the Google Drive, and we will we'll put a separate folder for those to.
Perhaps there could be some way of actually adding extra topics that are not included within the 4 topic groups to into the final
that
list.
That would be great.
Just because I think that the definitions of the topic groups and the way that we've come to this list might miss something.
Our goal here was to take an orthogonal view, but
Okay.
we were hoping everything would fit into one of these 4
Okay.
topics. So if that's really not working, we can make another topic, grab bag miscellaneous, but I think if it loosely fits into one of these, it might be easier for process.
Okay.
I think a lot is going to fall into topic 4,
Okay.
which are data sets, methodologies, technologies.
Okay.
So maybe that's the place. I don't know if Eve might get mad at me for saying that, but she's talking to somebody else right now, so we can just say that they're all going to go there. Carolyn, you wanted to make a suggestion?
Oh, no, I was just going to say, I agree. I think that there is the sort of, there is the doesn't fit clearly part, and so I think we can reconvene on that and make sure that that's getting covered and transferred into the sort of the synthesis and prioritization part. So we'll take a look at that and sort of call on the topic groups to make sure they are agreeing in terms of the things that we're doing with that.
Can I make one plea, because we're on it? For those of you, microphone. Sure. Yeah. For those of you who are seeing something that you think is really missing, please, I ask you to work hard to figure out which topic it would go in and what it's related to. Because what we want to avoid is minor, especially at this level that we need to look at things, we want to avoid having minor differences between things that are clearly quite related, because we have asked the topic people to try to condense. And so we push them to condense because we need to be able to get our heads around it. So we ask you to help them do that if you're going to suggest something that you really think is missing.
I did have a question, Adam. If we're actually going to be voting on priorities in some way, what we'd really like to see is that ballot an hour or two to sort of, so that's the stage where you want to know, is it there or not, right?
Yeah, so we'll try to create some time tomorrow when we have the lists from all the topic folks. But the way the agenda worked out, the people who went today have the luxury of dinner and breakfast to put things together. The folks that go tomorrow, please have mercy on them because they have a very short period of time to do that. But yes, the idea is that we're going to try to do it's a little experimental. We have to see how many different subjects we get to know exactly the details of how we're going to do it. But we do want a dot storm vote. I want to point out, reiterate what Mike Benke said. We are not going to use the results to say this got 8 votes, we're definitely doing, and this only got 4, we're definitely not doing it. No, we need some way to get a consensus from everybody and to help focus our attention on what needs development. And again, all this also will feed into additional planning processes, so it will all get mulled over. Elise, did you want to say something?
I was just going to say that, reiterate, we're saying exactly what you were going to say, this is not a binding vote. This is really to help give us some sense of prioritization. I also think that if there are certain topics that come up that you feel like we've just barely scratched the surface on that really need more in-depth discussion, please flag those for us. We can always have a follow-up 3-hour webinar on something. We don't, you can do it from a nice warm locale, whatever, but if there are things that you think need a lot more discussion, that we can do that.
And there are other things we're considering. For example, having a vote tomorrow, and then thinking about the results, and then later having a revote. There are other things that we want to do to try to help us prioritize these ideas and to help consolidate them.
Yeah. Yeah.
We start at 8 tomorrow. So.
And Adam, if people have specific questions, we'll be here and come up here. I just wanted to just take a minute here to thank you all. I know we're going to thank you again at the end of the day tomorrow, but I feel like at the end of day 2, it's a really good time to thank you all for the amount of time that you're spending here and the quality of the feedback and the input and the questions you're giving us. I can't stress enough how valuable I found this and how important I think these 2 days are, and keep it up into day 3. You know, I know it can get a little tiring, but it's really, I think, going to be another important day of the meeting. And so this is just my encouragement to stay strong and stay clever, and thank you all very much.
And I want to particularly thank the people who persevered through multiple attempts and cancellations of their travel due to weather and other things. So.